Okay, so welcome back. This is part two of the cell division lecture. And previously we talked about chromosome structure, and right now we are going to learn about mitosis. So mitosis is the actual act of dividing the nucleus. Okay, so this is distinct from cell division in that mitosis is just nuclear division. Okay, so in a cell, right, if we have a cell, we have a nucleus and we have cytoplasm, right? And so cell division means that we're going to have two cells that have all the same stuff. And so we have to divide both the nucleus and we have to divide the cytoplasm, right? And so the terms we use here are mitosis, which is nuclear division. The old days it used to be called karyokinesis. Remember karyo, like you carry it. Okay, karyo is that word for kernel. So karyokinesis was the division of the nucleus, as opposed to cytokinesis or cytokinesis, which is the rest of the cell. Okay, I don't know why we decided to start calling mitosis instead of karyokinesis. Karyokinesis and cytokinesis actually made a lot more sense in my brain. So anyway, mitosis plus cytokinesis gives us the M phase of the cell cycle, or we can now say mitotic cell division. Okay, all right. Now the opposite of mitosis or mitotic cell division is apoptosis. Apoptosis is pretty much ritualistic cellular suicide, as we said in the earlier part one. Okay, so where mitosis works to increase the number of cells of an organism, apoptosis decreases the number of cells. Okay, and apoptosis occurs at all phases of life. It occurs during development to remove unwanted tissue. Okay, so think of the tadpole's tail that gets removed. Think of the webbing between your fingers when you are a developing embryo into a fetus, and then your hands emerge from that. So the scaffolding breaks down. Apoptosis also plays key roles in our immune system, and it's happening right now all the time in your body, on your skin and in your endothelial cells. So your skin right now is shedding constantly. Your cheek cells are shedding constantly. Every second of every day, you're making about 100,000 cells, and about 100,000 cells are also dying through apoptosis. Okay, apoptosis also plays a very crucial role in preventing cancer, and we'll get to that later in the semester. And the way apoptosis works is pretty straightforward. Cells, in response to some stimulus to kill itself, whether it's an internal signal that happens inside the cell that says, hey, something's wrong in here, kill yourself, or there's some extrinsic signal that other cells are saying, hey, there's something wrong with you, kill yourself, the cells will eventually kill themselves. And this is a very carefully controlled mechanism through the action of proteins, including caspases. There's also ICE proteins, and there's death domain receptors. There's all these very heavy-duty sounding proteins that are involved in cells killing themselves. And what happens is the cell basically detaches from the rest of the tissues around it, the rest of the cells around it. It blebs up, it rounds up, the nucleus starts to fragment into these little pieces, and then the cell itself blebs and breaks apart into these things we call apoptotic bodies, which then get ingested by other cells like white blood cells. Okay, so you can see here in this little electron micrograph, you can see these are apoptotic bodies from the remnants of a cell that has died through this process. Okay, the next point is these things called cell cycle checkpoints. Okay, you can see these little stop signs here. There's one in G1, there's one in G2, there's one in mitosis. Okay, notice that there's none in S phase in this diagram, but in reality, there is an S phase checkpoint as well. Checkpoints are opportunities for the cell to stop and assess itself for damage. And if the cell is not healthy while it's going through interphase or through mitosis, it will instead shift gears and start the apoptotic programming and kill itself. Okay, this is essential to prevent cells from becoming cancer, because if a cell does divide when it shouldn't because of inherited mutations or acquired mutations, as those cells divide with the mutations, they can lead to cancer. Apoptosis prevents that. Okay, so we've covered apoptosis, we've covered the cell cycle, we've talked about chromosomes. Now we're going to get to the actual stages of nuclear division. Okay, it's all leading up to this point. You might remember some of these words from early grade school or high school, and the one new one here might be prometaphase. We'll talk about that later. But I'm also going to change the color of my pen at this point. Let me get this to be black. It's just easier on the eyes. All right, so I'm going to give you a very brief overview here of the stages of mitosis. Now, at this point in time, the cell has woken up. It's gone from cell cycle arrest, or G0, into G1. It's then moved into S phase. It moved into G2. Okay, and now it's getting ready for M phase. Okay, and so this is M phase. M phase is, here's your cytokinesis over here, and here's your mitosis over here. Okay, the first thing that happens when the cell leaves G2 and goes into mitosis is the nuclear chromosomes, okay, begin to condense. Okay, remember that genes are on chromosomes and that those chromosomes are in these tangled balls all over the place here. Okay, they start to compact themselves. They start packaging themselves for easy transport. Okay, at the same time, the nucleus itself begins to fragment or deteriorate. And so you could say that the nucleus starts to fade away, okay, as the chromosomes become more and more dense, more and more condensed. Now, at this point, in the rest of the cell, okay, if this is the rest of the cell, what's happening here is there are these pair of centrosomes. Okay, remember those are the microtubule organizing centers. And at this point in time, the centrosomes have a whole set of very organized microtubules called the spindle. Okay, and what the spindle does is it does a few things. First, we're going to anchor the centrosome in place. So it's fixed in space. It's anchored to the walls of the cell. And what's going to happen later on between these anchors and, ready, these microtubules, the ones that reach across the nucleus or span across the nucleus, these guys here are going to push away from each other. And what's going to happen over time is the cell is going to actually grow apart. And that's what's going to drive the process of cytokinesis, where we get the two cells. Okay, so let me start all that over again. Here's the nucleus as it's going to fragment. Here are the two centrosomes. Okay, the DNA starts to condense, and we have the spindle forming. Now, the spindle fibers I'm drawing now are all going to connect to the forming condensed chromosomes. Okay, and notice that each centrosome grabs onto one chromosome at a time. In reality, what's happening here is, if this is a chromosome, and here's one sister chromatid, and here's another sister chromatid, and here's a centrosome, and there's a centrosome, what's happening is the centrosome is grabbing onto the centromeres. Actually, it's called the kinetochore. There's a little protein structure on either side here. They're like tow hitches. So if this is a centromere, if I zoomed in here, there is this protein complex on either side called the kinetochore. The kinetochore, and the kinetochore attaches to the microtubules. And this will pry apart the sisters over time. Okay, so prophase, chromatin condensation, nuclear fragmentation, the formation of the spindles. That's what happens here. Okay, that's starting to what happened. Now, prometaphase is this intermediate phase that some books talk about and some books don't. Your book does. So the one thing that happens in prometaphase is the tug of war begins. Okay, so these are the two centrosomes, and the centrosomes are tugging here on that chromosome, and they're tugging. Let's say there's a chromosome over here somewhere. Okay, he's tugging on this chromosome over here, and let's say there's another chromosome over here somewhere. Okay, and he's tugging over here, and let's say there's another small chromosome over here. Okay, so we have all these chromosomes. Notice they're all over the place. Okay, so what happens is there's a tug of war that commences where this centromere pulls this way and this centromere pulls this way, so that over time what happens is the sister chromatids, the chromosomes, are all lined up in the center of this tug of war where nobody wins. Okay, so this lining up when all of the chromosomes are in the middle of the cell looks like this. Okay, basically all the chromosomes are in there. When they're all lined up in that middle, that's metaphase, the middle phase. Okay, and this area here is called the metaphase plate. Okay, it's not a line. Remember that cells are three-dimensional. So if this is a three-dimensional cell here, we're on 3 dimensions here, the metaphase plate is this two-dimensional plane that's in the middle of the cell. And all along that surface, you have the chromosomes being separated by, or being pulled rather, by the 2 centromeres. Okay? All right, so here we go. That's the cell. There's a nucleus starting to condense. And we got the centrosomes on either side. Prometaphase, we have the, let me erase, you know what, sorry, let me do that again. Here's the nucleus, here's a cell without a nucleus, right? And so we just have these condensed chromosomes everywhere, and we have the spindle that is formed, okay? And then the tug of war is over here. This is the beginning of the tug of war. And then in metaphase, we have this beautiful, elegant, everything lined up in the middle, okay? The next phase occurs when the sister chromatid are separated. So in metaphase, the chromosomes look like this, right? And they're being pried apart. In anaphase, the cell itself starts to elongate as the cell comes apart. The centrosomes migrate towards either side of the cell. And what happens is the spindle pulls the chromosomes apart. Okay, pulls the sister chromatids apart. So that's anaphase. At this point in time, notice that cytokinesis also starts. Cytokinesis is where the rest of the cell starts to divide. So what happens here and here is the cell begins to pinch in a little bit. Okay, and the reason for that is there's this structure called the contractile ring that forms around here. It's made of actin. That's the contractile ring. It's made of actin. Sorry for the sloppy handwriting. And the actual shape of this is called the cleavage furrow. Okay, so the cleavage furrow begins in anaphase, and this is the beginning of cytokinesis. So all the cell's contents are going to either move to the left or move to the right, along with the nucleus that's going to beginning to develop here. Okay, all right, so once the sisters here are separated from each other, telophase is this final phase of the cell cycle. I'm sorry, final phase of mitosis, where you have the beginnings of the 2 cells now. You can see the 2 daughter cells taking shape. Cytokinesis continues, and the nucleus reforms. And so the nuclear membrane reestablishes itself, the chromosomes decondense, and you get 2 nuclei. Cytokinesis continues through telophase. Notice that it just keeps going here. Okay, and actually it doesn't really complete until the next G1. Okay, and this region here is called the mid-body. You don't really need to know that, but that's what that is called. Okay, so that is mitosis in a nutshell. Okay, prophase is when the chromatin condenses and the nucleus fragments. Prometaphase is when the tug of war begins between the 2 centrosomes. Metaphase is when you have all the chromosomes lined up on the metaphase plate. And I should note here that when they do line up on the metaphase plate, there is no particular order. All the chromosomes are randomly jumbled there. It doesn't matter what their order is because each cell, when it's done, is going to get exactly the same thing. So it doesn't matter what order they're arranged in there. Okay, anaphase, the sisters separate, and in telophase, the nucleus reforms. Okay, so those are the stages of mitosis in a nutshell. Now the rest of these slides basically dig into the details of each of the stages. So let's just go through these and clarify anything that I missed. All right, so in prophase, as we said, the nuclear membrane disappears. Centrosomes migrate. What that means is the centrosomes, if that's the cell and that's the nucleus, here's the centrosome, it duplicates. So you get two now, and the other one migrates to the other side of the nucleus. And at that point, the spindle appears. Spindle fibers appear. Okay, the spindle grows from between the centrosomes. So if we take two centrosomes, the spindle forms between them, and the reason why they push apart is because these microtubules are actually doing the pushing. Okay, there's little motors in there that push these things apart. Okay, we talk about chromatin condensation. So this is the word that means the DNA is just getting tighter and tighter and tighter and denser until we can actually see it under the microscope, as you saw in lab, or some of you saw in lab. And so basically the chromosomes become visible. And at this point in time, you can see, usually you can see the two distinct sister chromatids, right? So remember that a chromosome can be made out of one chromatid, or we just call it a chromosome, or after S phase and it's duplicated, we have two sister chromatids. Each one of these things is a chromosome. There's one chromosome, and the way you count chromosomes is you can just count the centromeres. Okay. All right. So, and then the centromeres attach to spindle fibers. Okay, rather not that chromosome, but this chromosome attaches to the spindle during prophase. Okay, and the key here is that the chromosomes don't really have any particular orientation in space. They're pretty much all over the place. During prometaphase, those centromeres begin moving away from each other towards opposite ends of the nucleus. Okay, so it's a little subtle at this point in time, but eventually we can see those two centromeres are literally moving away from each other, and the cell is slowly starting to get longer and longer and longer. Okay, this way, it's starting to get longer as it gets pulled apart, and the centromeres control this. Okay, so that's prometaphase. Here are some pictures of the processes. So this slide here shows early prophase, prophase itself, and prometaphase. So in early prophase, you can see the nucleus is starting to break down. The chromosomes are decondensing. I didn't really talk much about this, but the nucleolus disappears, which makes sense because it's made out of chromosome. It's made out of bundled chromatin. You can see here the pair of centrosomes migrating away from each other. Okay, in the center here you can see the centrioles. Remember that only animal cells have those. A couple plant cells do, but only ones with flagella, and you don't have to worry about that. Okay, plant cells, for your intents and purposes, plant cells do not have centrioles. Animal cells do. Okay, during prophase, again, you can see here the centrosomes are migrating apart. You can see the duplicated chromosomes. Okay, it's a big tangled ball here, right? So here's a centrosome, here's a centrosome, here's the spindle. You can see the spindle everywhere here. Okay, and you can see that a lot of the spindle is kind of embedded in this messy tangle of chromosomes. All right, but notice it's still messy. Okay, at this point you can see, because it's still pretty intact, you can see the nucleus itself still. Okay, and then notice here there's no more nuclear membrane. Okay. During metaphase, the spindle fully forms and consists of poles, asters, and fibers. An aster we didn't talk about before. An aster is just a spindle fiber that is not attached to a chromosome. Okay, so these are asters. Okay, these are asters. And then these are spindle fibers, okay, that are attached to chromosomes. And at this point in time, the chromosomes line up at the metaphase plate. They're all in the center. And then during anaphase, they get pulled apart. Another way of saying anaphase is that the centromeres divide. Okay, so one duplicated chromosome becomes two grown up chromosomes. And so you can think of this as we went from one centromere to two centromeres. Okay, so the centromeres divide. Sister chromatids are then moved to opposite poles by the fibers. Okay. And then during telophase, the nuclear membrane reforms, the spindle disappears, and cytokinesis, I want to say cytokinesis continues. So really, let's go back a slide here. The key here also that's missing is that during anaphase, cytokinesis begins. And that includes the formation of the contractile ring and the formation of the cleavage furrow. Okay. All right. And then telophase, you could also say that telophase is the opposite of prophase. Okay, so everything that happened in prophase goes backwards now. The nucleus, instead of fragmenting, reforms. The chromosomes, instead of condensing, decondense. The nucleolus reappears, and we have two daughter nuclei. Here are the pictures of those three phases now. Metaphase, you can see very clearly. You can see that all the chromosomes are lined up in the middle there. You can see the plate. You draw a line like that. Okay, so that's metaphase. And then just as clearly, you can see in anaphase, you can see the separation of the daughters getting pulled apart. Also notice that the cell itself is getting a little longer now. It's getting more oblong, more like a football shape. Okay, and by telophase, you can see that there's two cells. You can even see in this picture here, if I erase all that drawing, you can see the contractile ring and the cleavage furrow. So there's a cleavage furrow, there's a cleavage furrow, and then what's not really discernible here is there's a contractile ring around here. Okay. All right, let's see, any other details that I should warrant here? So if you look at the diagrams down here, you can see the little yellow dots, those are your kinetochores. You can see the spindle fibers attached to them pulling in opposite directions. You can see here other spindles that we call asters. These are anchoring these things in place. Okay, you can also note here that there are these are called polar spindle fibers. You don't need to know this, but these are the driving force behind the force that pushes the cell apart. There's little motors in between these guys that are pushing everything away from each other, and that's how the cells separate. Okay, that's pretty cool. All right, and then you can see in telophase, you can see the nuclear envelopes reforming. You can see the chromatin begin to decondense, although they don't really draw that here very well. And you can see here that each cell also has its own centrosome and pericentrioles. All right, so that's animal cell mitosis. In plants, it's pretty much the same thing. Okay, there's a few minor differences here. And you don't need to know this. I'm not sure why I really have this in this presentation, but the meristem within plant tissue is the part that is growing as opposed to the part that is done growing and differentiated. Okay, and the other key here is that there are no centrioles and there are no asters. Okay, and the reason for that is the cell will not be going through the same kind of cleavage furrow and contractile ring and that kind of pulling apart. You'll see a completely different mechanism here. Okay, so in plants, you can see prophase to prometaphase. Okay, you can see the chromatins condensing. In prophase itself, you can see individual chromosomes, but they are chaotic, they're disorganized. All right, in prometaphase, you can see that the, well, really what's neat is you can see that there's no centrioles and there's no asters. Okay, in metaphase, you can see a metaphase plate, just like in animal cells. And in anaphase, you can see the daughters being pulled apart. Now in telophase, notice there's no contractile ring here. Okay, there's no loop here of actin. Instead, what happens is each side of the cell, which now has its own Golgi apparatus, begins to basically throw vesicles across from each other, and those vesicles line up in the middle and form these little droplet shapes that eventually form a new cell wall. And you can see that beginning to form right here. Okay, and this thing is called the cell plate. All right, so you can see the cell plate pretty clearly there. Okay, so this slide just summarizes the cytokinesis in animal cells. Okay, so the cleavage furrow forms between the two daughter nuclei. The contractile ring contracts, it deepens the furrow, and the process keeps going until the cell is completely separated from each other. Okay, so you can see another cell here going through cytokinesis. It's an animal cell. Okay, and on plant cells, cytokinesis requires a new cell wall between the daughter cells, right? And so a flattened small disc shows up between the two daughter cells, and this begins to form what's called the cell plate. And the vesicles along that cell plate release molecules that build new cell walls. And the vesicles themselves become new plasma membrane. Okay, so here you can see, here's the cytoplasmic side of one cell, and the cytoplasmic side on the other is over here. Okay, I'm sorry, let me redo that. I misspoke. The cell plate is right here, and so this is one cell, and this is another cell down here. So let me do a little schematic here. If we were going to continue, here's the rest of the cell. Okay, so here, up here, this is cell number 1, and down here is cell number 2. Okay, and so this thing across here, that's the cell plate, and this is the cell membrane. Okay, so you can now see clearly. Here's the cell membrane here and here, right? And then these little vesicles all along here, you can see the beginning of the cell plate. So these vesicles will, inside them, form the material necessary for the new cell wall that will be basically building out like that. Okay, you can see the old cell wall here. Excuse me. And then the outside of these vesicles will form the plasma membrane. Okay, so here's a summary of the stages of mitosis. This is just for your own studying, a different way of looking at everything. Okay, notice there's interphase here in this slide. So interphase includes G1, S, and G2. Okay, so interphase occurs, and then prophase, prometaphase, metaphase, anaphase, telophase. And there we go. And so you can kind of jump back and forth between these two slides, and you can see the pictures of these. Okay, it's pretty cool. So in these cells, you can see during prophase, you can still see the nucleus. Okay, in prometaphase, the nucleus is beginning to go away. The red hairy things, that's your spindle. You can see in metaphase a beautiful lined up plate. And then in anaphase, you can see them all getting pulled apart. And during telophase, you can see the beginnings of the new nuclei. Okay. Okay, more slides for your studying. You can use the PowerPoint and focus on those. I like showing students many different ways to do the same thing. So on the top here, you can see the blue corresponds to the nucleus. You can see this green glowy thing here. This represents the centrosome. Okay, so you can see the centrosome is always right next to the nucleus. Okay, and over here you can see there's one and there's the other. Okay, there's metaphase. In this case, you can see a whole bunch of chromosomes. These pictures might actually be cancer cells because there's just so many chromosomes here. Most cancer cells have way more chromosomes than they're supposed to. There's just so much wrong with cancer cells. Don't even get me started. Right, and then here's anaphase. You can see the separation of all these chromosomes. Okay, and here's cytokinesis. You can begin to see the mid-body forming. You can see the cleavage furrow pretty clearly. You can see that the chromosomes are already decondensing. And over here in the neighboring cell, here's a cell that hasn't gone through mitosis. So you can see a very nice intact nucleus and the rest of the cytoplasm there. Okay. All right. and more slides, more pictures of these things. So I'm just going to gloss over these. Okay, these just show you all the different steps again. Okay, and this website here has a really good slideshow of the different stages of mitosis. All right, so I'm going to stop there. That concludes part 2. We've just covered mitosis, and in the third and final part, we're going to cover meiosis.